Okay, so good evening. Welcome to week three of Introduction to Proteomics course. So before that, before we dive into the course, can you all just recall what we did in last week? What were the major points we covered in terms of last week's lecture? Yes, yes. Can you recall, I Siri? What did we do last week? Yeah, great. So basically, we done the initial step with before doing the analysis. So how do we exactly make prepare samples and what all precautions we need to take care, and depending upon the type of analysis, type of sample, the processing will change, like the sample preparation will change. So we discussed few things related to that, and we did one of the analysis method, which is SDS page, which only by one dimension, and we also try to do a bit of introduction to two-dimensional electrophoresis. And this week we'll be discussing in detail about the same thing about a two-dimensional gel electrophoresis. So before that, maybe and like you can unmute yourself and ask if you have any doubts while I'm taking the lecture. So yeah, welcome to this week three lecture of Introduction to Proteomics. This is an NPTEL course which has a code of BT60. And week three will be majorly dealing with this specific technique, which is two-dimensional gel electrophoresis, also abbreviated as 2DE. So what we'll be doing in this today's lecture is we'll be talking about two-dimensional relative versus the second dimension. The first dimension we did in week two itself, that is isoelectric focusing, and after that there's a step called equilibration, and we talked about that too. In week three, we'll be starting with the second dimension, that is SGS page, how we can do it, staining, destaining, etc. Then how we can analyze the data which we obtain and different applications of the same of this specific uh, technique which can be used for different applications and certain challenges associated with this specific technique. And uh, there was a lab session as well of analysis, uh, as well as how this isoelectric focusing gels are prepared, etc. So you might have gone through that, so that would be helpful for you all to understand how exactly it is actually performed in the lab. So this was a good session. You can go through that. So I'll be majorly covering this theoretical part. So just to give an overview what 2DE is. So initially you run the first dimension, which is isoelectric focusing, wherein you run the samples on the basis of pH grade, like there's a pH gradient strip, and depending upon the PI, what is PI basically? PI is nothing but a point at which protein achieves net zero charge. So depending upon the type of protein, it has different PI values, and it will stop moving at that particular point because it achieves zero charge, and it as it is zero charge, it won't be affected by plus and minus charge here, and it will stop moving. And that's how the separation takes place here. Then after that, there's a step called equilibration, wherein disulfide bonds are broken down and alkylated as well, so that it doesn't get reformed. And after that, they are subjected to second dimension, which is SGS page. Now here we don't require stacking gel. The proteins are already stacked. We just require dissolving gel, and we can directly separate them on SGS page. Now here the separation is on the basis of molecular weight. The higher molecular weight are above, and the lower molecular weight, like they run faster, and that's how separation can be achieved. So this technique is basically two-dimensional electrophoresis. We've already gone through the first dimension one in our week two lecture. We'll be talking about the second dimension, how exactly that can be performed. And after it is performed, what all different types of staining techniques are available. We'll be talking about that as well. So just to give a practical picture how exactly gel is casted in terms of uh, SJS page and isoelectric focusing, this is how the gel looks like. Uh, so in terms of rehydration step or equilibration step, all that can be done in a soaking tank. It looks something like this, and you can place your strip in between these tank and add that specific buffers and equilibrate it or rehydrate it accordingly. When you run the SDS page, uh, this is how your isolatory focusing strip is kept, and it will accordingly run the sample. This is how the tank looks like in terms of SDS page. What does SDS do to the proteins? It generally gives negative charge to the protein. We have already discussed the one dimension uh, assay, which was SDS page. So we are doing the same thing. How exactly those gels are prepared using TIMED, APS, acrylamide, bisacrylamide. So we have already gone through that. So we are not going in detail here. So that's the same technique we are repeating after isoelectric focusing. Separation happens on the basis of molecular weight, and so that's what SDS page is. That's what second dimension is, wherein proteins are separated on the basis of molecular weight. We don't require stacking gel here in second dimension because the stacking is already done by isoelectric focusing step. And once that is done, now you can stain and destaining method. It depends upon what type of protein you are targeting, how much concentrated protein you are targeting, and what type of visualization technique you will be using further. And if you are compatible with mass spec, if you are not compatible with mass spec, so if you are aiming to do mass spectrometry later, accordingly, those stains need to be avoided which are not compatible, and whichever are compatible accordingly you can use those. And also depending upon the concentration you wanted to check, lesser concentration, or for example you wanted to check any post translation modification modified proteins, then different stains are available accordingly, and accordingly to your aim or according to your interest of protein which you want to detect, that type of staining technique can be used. So one of the most commonly used technique is comassive blue stain. This There are different stains like comassive blue as well as biosafe comassive stain. Types of stains are available, and it is commercially available and it is ready to use. And it's basically a single reagent protein stain. And this used stain can be disposed as non-hazardous waste as well. That's the benefit of biosafe comassive stain. So how do we stain it? Basically, after running the gel, you remove the gel from the plates and you add it. You take a plate or something like that which can hold the stain, and the staining can be done overnight as well, depending upon the type of protein, source of protein, etc. Some proteins might require overnight staining, and some can be done like 2 hours also. Like it depends. Any technique can be used. That's one thing. Comassive blue staining. Then if you want a more sensitive technique like which can detect even 1 nanogram of protein, because comassive can detect around 40-50 nanograms, it cannot detect lesser than that. If your protein is very less concentrated and you want to detect them, then silver stain is the thing which can be used. So in order to do that, you need to fix the proteins first by using methanol and acetic acid. Then you can add the stain. Then this specific silver staining has a complex between tungsten silicic acid and silver amine. So in what happens in this, AG ions transfer from this specific silicic acid to proteins, and that's how staining takes place. Then the silver ions are also reduced to metallic silver, and that's what helps in visualization later. And this specific reaction is stopped by adding acetic acid. So in terms of silver staining, you need to add that silver stain onto the gel, and that specific reaction takes place. This which we were talking about, AG ions transfer this tungstic acid to proteins, and that reaction is stopped by adding acetic acid, and later it is visualized uh, under UV light itself. There are other types of stain as well, like cyproruby, which is a fluorescent stain, and it is visualized under UV, and it gives little background. It is much more sensitive, and it's basically an endpoint stain. And one of the benefit of using this is it can also detect post-translated modified proteins, like it can detect glycoproteins as well. And it requires even low molecular weight proteins are much more easily detectable with cyproruby, and it is compatible with mass spec too. Endpoint stain indicating, even I'm not exactly sure about it. Uh, I'll get back to you related to that. Even I'm not sure, like I don't want to give any random answers to your question. So even I will have to go through that exactly what that exactly means. Yeah. So before staining, there is a fixing solution in terms of silver stains. Like for few stains, you initially require fixing, which has methanol and acetic acid, which fixes the protein so that it gets compatible with the stain, so that it can take up the stain and uh, like basically the protein will be stained because of the fixation. So that's generally required for few of the stains and not for all the stains. Then after that, in order to stop the reaction, the percentage gets reduced, methanol and 7% acetic acid, which is also called as washing solution, or this can be also called as destaining solution. So that incubation can happen for like 1 or 2 hours, depending upon the type of protein. Once that is done, you will remove that and you will add a washing solution, which can be also called as destaining solution, and that has 10% methanol and 7% acetic acid. And after destaining, so why we require destaining? Because generally, for example, if this is a protein gel, it initial staining step, when you're staining it stains the whole gel, and it stains the protein much more intensely, but it can stain the whole gel itself. So destaining is required in order to remove the stain from the gel part, and only proteins which are actually present, only that specific protein will be stained, whereas the gel won't be stained as such because of the destaining step. So destaining, that's the reason destaining needs to be done. So, if it is not done, then the protein as well as gel will everything will be stained. So you won't be able to detect spots of the protein. So destaining is required. That's why, and so that you will be able to detect protein spots. And so depending upon the type of protein, as this is a fluorescent protein, so you require certain excitation and emission wavelength. So at that specific excitation wavelength and emission wavelength, you need to set that specific protocol, and you can visualize this specific gel. There are other types of stain as well, which is diamond, which is a fluorescent dye again, and it detects phosphorylation. So again, in terms of cyprobrew, it can detect glyco proteins, whereas this can detect phosphoproteins. So depending upon your type of protein you want to detect, if you want to see some post-translated modified proteins, you can either use Cypro or ProQ. You can use combination of them as well, and this is much more sensitive, can detect nanogram levels too. It's the same thing, fixing and then staining and then washing. And it has a different excitation and emission wavelength in terms of ProQ diamond. So you can do dual staining as well, wherein you can mix both of them, ProQ diamond and cypropro ruby, where you can detect both glycoprotein as well as phosphoproteins. So if you see all of this, most of them are compatible with mass spec as well, MS mass spec compatible. There's only silver stain which has some compatibility issues, whereas all other stains are generally compatible with the mass spec analysis. So if you're dealing with any If you're planning to do mass spec after your SDS page, then make sure that you use the one which is compatible with mass spec and not Silverstein, which is not compatible with mass spec. But if you are just aiming to check the protein and you are not planning to do mass spec, you can obviously use them because it can detect even 1 nanogram level of protein, which is highly sensitive technique. So using this would be beneficial if you just want to do SDS page as compared to that of for massive blue because it detects 40 nanogram or 10 nanogram of protein. So this is what which was discussed in this lecture, and then sir also gave few of the references. You all can check that for knowing more details about the SDS page. Then now you have run the sample. How you can analyze them in terms of the same 2D gel into how what are the ways by which you can analyze and what applications this specific technique can be used for. So for imaging, there are different tools available. One of it is Typhoon Variable Mode Imager and as well as Molecular Imager, which is also a densitometer. So densitometer indicating it can tell you about the density of that specific protein in that lane or in the whole, like in that specific well of the gel or else the whole gel, depending upon the type of analysis you are doing. So densitometry indicating, for example, you added a lysate, like you have taken the cell and you are trying to check one of the protein which might be produced, which might be expressing in that specific cell. You will lyse the cell, you will do fractionation, everything is done, and then you are loading the lysate itself on SDS page and checking whether your protein of interest is there or not. Even if it is there, how much it is there, like how much percent. Like you can do a relative study using densitometer. So, for example, you added the lysate and your protein of interest is giving much more denser band as compared to other proteins. So that density can be determined by comparing with the whole lane, and it can tell you that 50% of the protein is your protein and another 50% is all other proteins of that cell. Sometimes it can be 30%, it can be 70%, depends. Like if the protein is much more overexpressed because of engineering the strain, etc., then you will able to detect that much more intense band and it will tell you about the density present as compared to the all other proteins present inside cell. So that's what densitometer can do. Even Typhoon variable mode imager, it helps you for much more better analysis, like something like this. So it can help you to spot identification. It can help in comparison of gels. It can help in overlaying of images. It can also tell you give cropping gels as well. Like instead of determining the whole gel, you can just crop it the interest. Like if you know the molecular weight, if you know the isoelectric point, you know where it might be your protein of interest. You can just zoom into that and detect only that part of do analysis of that part of the gel instead of analyzing the whole gel because it will have a lot of proteins and your analysis might be much more difficult. Like it allows to do cropping gels as well and becomes so that analysis becomes much easier of your protein of interest. So what does crop tool do? It allows specific defined region, which we just discussed. So specific region can be detect like cropped and only that part can be taken further for analysis. Zoom tools can be done like it can expand a specific area for further analysis. For example, if two proteins are very close to each other, like if it is a zoom out image, you are not able to detect whether they are separated or not, and what might be the exact molecular weight of those two proteins which are close by each other. So zooming can help you all to separate, like detect each spot separately and help in analysis of those two proteins. So that's what zoom tool can help you all in terms of image overlaying. So one image like maybe healthy control was run in one gel and disease control was run in another gel. When you overlaid, you found that if blue and yellow coming together, it forms green. So it says that even so these are the proteins which are present in healthy as well as disease patient, whereas this is specifically present in healthy patient, but it is not present in disease patient, whereas these proteins were present in disease patient only and not the healthy one. So That's how overlaying will help you to compare spot patterns of two different gels. For example, so it depends what type of gels you are running and that type of analysis you will be doing and coming to certain conclusions accordingly. So spots that can coincide lie on top of each other while others retain their original position. That's what image overlaying will help you all to do certain analysis according to the aim. Then it can also tell you about the intensity. So if that specific protein is much more intense or less intense, can all be done like using statistical parameters. So it allows comparison of comparison spot by spot basis. So you can actually detect each and every spot separately and also comment upon whether this is more intense compared to this or this is more intense compared to this. Even comparison can be done between two gels. All such type of analysis can be done. So, for example, control versus treatment, these are two just healthy control and disease control. It shows that these few proteins were much more overexpressed in healthy compared to that of the diseased one, whereas you can see few of the proteins here which are not present here, like this specific protein, or these are much more overexpressed here compared to the healthy control. So all that type of analysis, just by looking at the uh, gel here, you might not be able to understand just by looking, but your software, when you are doing dealing with software, trying to compare each, so that's what it will help you all. Like it can detect each and every spot separately. Like if they are separated, it can zoom in and check whether they are separated or not. All that can be done with the help of software. And so sir has also given like few links for different softwares. If you are willing to go through it, you can go through that. It can help in gel matching, give certain scores to it, how much it is overexpressed or how much it is downregulated or upregulated, etc. Such type of analysis can be done. It gives identity to each and every spot. Like it will give some name to it, like an ID to it. It tells you about the intensity of each and every spot, area of that spot, volume which might be present, etc. So all this can be done just by looking at the gel like this. Like the software will be able to tell you uh, everything which we just talked about by looking at one spot itself. So it can also tell like if there is any specific protein which is not present in the disease one but present in the control one, etc. All those can be done. It can tell you about the PI like because initially it run in isoelectric focusing. It will tell you about the PI. It will tell you about the molecular weight. It will about tell you about the spot area, how much it is, the volume, intensity of that specific spot. Everything can be detected, analyzed basically by using this software. So that's how you do analysis. Uh, so it can help you to study the global protein expression. If you're studying the whole gel itself, globe like all the proteins, that global expression. Study differential protein expression, indicating you have run the healthy one, you have run the diseased one, and trying to study how different they both are. That differential protein expression study. Resolve proteins from complex mixtures. That's obvious. Like that's what the technique is all about by resolving the protein. It can tell you about post-translation modification, isoforms, etc. And basically, it will help in visually analyzing the proteins. In your sample, it's just there. But when you do this technique, it will help in visual analysis of proteins. So, for example, there's condition A, condition B. You have run isoelectric focus in condition A, condition B. You have run the second dimension and you have stained it with the type protein you are interested in. Accordingly, you have used some type of staining technique. Now, in condition A, condition B, and they found that these were the few proteins which were not present in condition A. So, it's just talked about that condition B has some proteins which are not able to express in condition A. So you can study different conditions also. This can be one application, like for example, you're growing cells in one specific media and checking in another specific media and checking what type of protein changes is happening. Like whether you are supplementing certain uh, carbon source or protein source, etc., nitrogen source, etc., in condition A, but not in condition B. But if there is any changes happening. Like I'm just giving an example. So all that those can be studied at a protein level if there is any change happening. Like you can visually see it if there is any changes happening, or you can check by OD if there is because of certain supplementation whether the growth is much faster or lower. All those can be done. But if you want to understand at a protein level, then you can do this specific technique and understand it. So it's the same thing talking about the intensity, etc. So it helps in systematic separation, identification, and quantification of proteins. So if you have standard, you can exactly quantify, but you can do relative quantification, like this is higher than that. That's what you can do, like by using this. And if you have standard and if you have loaded a specific concentration and then compare, you can exactly quantify as well. It helps in analysis of cell differentiation, assist in detecting and identification biomarkers. Like basically, if this is different, then you can able to detect the biomarkers as well, and it helps in protein purification obviously. Then those were some analysis technique and one of the application. Then sir talked about few more applications related to two-dimensional gel electrophoresis. I hope you all are getting it, and you are, yes, everything good till now. Yes. No, we are not able to retain that because we are denaturing it. So if your aim is to study the activity and stuff, then you never do SGS page. So SGS page is just to detect whether your protein is there or not. But if you are doing activity, then you will lyse the cell and try and check the activity from the lysate. That's what you will be able to do it, like from the lysate. So or else, if you want to purify that protein and check the activity, you will directly take that lysate, do fractionation, like do affinity chromatography or gel filtration chromatography, separate your protein of interest is if it is that is purified. If not, then you do certain precipitation technique, dialysis technique, etc. Concentrate your protein and then check the activity. So in all this process, you're not denaturing your protein, so it's your protein is still active. But as soon as you subject to SGS page, it gets denatured. I didn't get your question. Yeah. Okay. 2D agarose generator, for instance, that is age, right? Like you're talking about. So age cannot separate your proteins. No, 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 no. So age is specifically for DNA and RNA. It won't be able to separate your proteins because proteins are larger molecules, and agarose will make much more complex gel, and proteins will not be able to get separated from age. Native page, you can use native page, although it still uses like, but why do you like if you are doing SDS and if you are subjecting to mass spec later, your activity is any which was lost, like in mass spec. I'm not trying. I'm trying to understand why do you want to maintain the activity and do mass spec? Like, why is that so? Mass spec will any which ways kill the activity, right? It will get fragmented. The whole protein will get fragmented. So mass spec, how does mass spec detect your protein basically? So for example, your protein is 200 kilodalton. So there will be n number of proteins which will be of 200 kilodaltons. But how will be exactly detecting that specific your protein? Like you have separated your protein on SGS page, there is 200 kilodalton protein band you see. but you are not sure whether it is your protein or not. It can be any other protein of the same molecular weight. So how do we detect that? How mass spec helps you for detecting that? So you take that 200 kilodalton protein, you just cut that specific gel, and initial thing is you subject to trypsin digestion itself. So you are digesting your protein in the first, like in the sample preparation step itself, where you are completely digesting your protein, like 200 kilodalton, like it might be some indicating there will be some number of amino acids, like you need to divide it by 3 accordingly. 110. So divide by 110, so you'll be able to get the number of amino acids present. So how does trypsin do the digestion, if you know? Like where does it cleaves the bonds? Do you know that? So basically what trypsin does is wherever arginine and lysine are present in the whole sequence together, like arginine then lysine, it will cut there. It will break that specific bond, break that peptide bond basically, and it will make different peptides of that specific protein. So each protein will have its unique sequence, and those unique peptides will be detected by mass spec basically, and that's how you will be able to correlate and identify like it was your protein of interest itself and it was not something else, because each protein has its own unique sequence, and it what mass spec will do, it will detect those peptides after arginine and lysine. You can do different types of digestion basically. Most commonly used is trypsin digestion. So those peptides will be detected by mass spec depending upon the mass and the charge to that specific peptide, and it will compare it with the library and check whether that's what the sequence of so each peptide will have its own molecular weight, and accordingly, that peak will be observed at at that specific molecular weight, because it separates on the basis of mass to charge ratio, right? Mass spec. So accordingly, it will be getting eluted at that specific time point, and you will be able to detect those peptides accordingly by comparing with the library, and that's how you detect whether that peptide is matching with your protein of interest or not, and that's what will tell you that your protein is present or it was something else at the same molecular weight. Getting it? Yeah. So you were interested in terms of activity. So for example, if there is an enzyme, uh, so you do enzyme activity assays and stuff. So that can be done like after purified protein itself. Like you do certain type of chromatography, you take the lysate, do certain type of chromatography, purify your protein. You have your purified protein, and then you can check its activity. Whatever assay you want to do related to that specific protein, you can do that. So that will tell you whether your protein is active or not active, etc. So that is different, like just checking the activity, but this is further much more accurate confirmation that that's the protein you were looking at, and that is the same protein which is giving you the activity, and all of those can be done with this specific. So it depends upon your aim. Your aim is related to just checking the activity, then you know there's no need of doing mass spec and stuff because it is giving activity indicating that your protein is present obviously. Yeah. Okay. So few applications related to two-dimensional electrophoresis. So do so one of the application was serum proteomic analysis of Vivax malaria. So there is one people suffering from malaria and people who are healthy control. So they try to check. So this is Vivax malarial sample, healthy control sample. So this is similar to that of condition A, condition B. Condition A, people who are having malaria. Condition B is healthy control, etc. Similar types of analysis, two-dimensional electrophoresis done. First isoelectric focusing, then to, SDS page, and then did the analysis, etc. And they found few spots. So in terms of serum, we did learn about it during sample preparation. So if few of them are highly abundant, so you just need to remove them first by affinity column chromatography or ultrafiltration, etc. So once you remove them, you will be able to detect these lesser amount or basically which are present in less concentration. So the detection range will be stable in order to detect these as well. So once you remove the highly concentrated proteins. So they have done some sample preparation optimization also in this study. So they have used the serum directly. They have desalted the protein and then checked. They have sonicated and did desalting, and they initially removed the abundant protein and then did the desalting. So they tried to check the sample preparation, how it can affect your results in the next stage. So this is just the protocol, how they prepared the sample, incubated it, centrifuged it, precipitated it using acetone. So we did talk about sample preparation. So they have used this specific method, and they found that the number of spots indicating after two-dimensional electrophoresis, after the separation, the number of proteins which were observed. So they found that just by using this crude serum itself, a lesser number of proteins were detected as compared to that of one which was sonicated, desalted, and depleted, indicating albumin and IgG were removed, and then the samples were analyzed. So it detected much more higher number of proteins, indicating that the smaller concentrated proteins were much more detected here, whereas they were not detected here because of the inhibition from albumin and immunoglobulin. So these were the few papers of the same, like they tried to study that, and they found that uh, by removal of abundant protein, it detects much more higher number of proteins. So this is how the gel looks like. Uh, so they have optimized the whole sample preparation. So if you are precipitating, you're doing protein fractionation using acetone or with this TC and acetone together. So this gave much more higher number of spots compared to that of just acetone. So this is giving more number of spots here, clearly separated ones compared to just by acetone. Similarly, crude serum versus depleted serum. So because of the presence of albumin, it inhibited the separation during the isoelectric focusing as well as during the SGS page. So that's the reason number of spots were lesser in crude serum compared to that of depleted serum. So if you can see, more number of spots were found. Similarly, type of staining technique you are using, comacid blue staining or silver staining. So silver staining can detect even like less than 30 nanogram, it can detect 1 nanograms as well. So it detected 995 spots compared to 919 spots in comacid blue. So there were around 50 to 70 proteins which were much more lesser concentrated, which were detected by silver staining and not by comacid blue. So the protein loaded in these two techniques was this much. So it's just a comparison, like a number of spots identified by different techniques, just by using crude or by changing the precipitation technique or the trizol method. So we also studied about trizol method of one of the separation technique and lysing by sonication and desalting, etc. So it changed the number of spots accordingly detected by the software. So this that's what it comes to be to, like the sample preparation is most essential and it can change your results drastically. There was one more, so this is the same study. So there were eight samples of malarial sample and eight healthy control samples, and they found that this was the difference between the healthy and the uh, malarial one. So you can see there were a few proteins here which were not present, but here it is present. Similarly, this is over upregulated compared to the healthy control here. Similarly, this is also much more intense as compared to that of this protein. Similarly, this protein in healthy control is much more stronger compared to uh, the malarial patient. So protein detection were done. So they found few of the proteins which were present in the malarial patient, but not in the healthy control patients. So all those can be detected. And if you want to analyze what protein is this exactly, then you need to do mass spec. It can tell you whether it is much more intense or less. Like in some healthy control, few of the proteins were upregulated, few were downregulated. Here in healthy control. Similarly, those proteins were upregulated in the plasmodium one. So all the statistical analysis can be done. So they found that few of the number of spots upregulated fold change as compared to that of the healthy control. So this was the range, like these many number were upregulated accordingly. This was the number of spots found. Then if you want to detect the exact protein, for example, if BS is there, then you can make standards of that and accordingly compare that with your analysis. And if you can see the similar peptides here, and then you can determine your protein of interest is present. So after doing all those types of analysis, they found that these were the few proteins which were downregulated, and these were few proteins which were upregulated in the malarial patients, and few references of the same. Then along with that, two applications, condition A, condition B, and malarial patient. Sir gave few more applications, like you can also study effect of drug uh, treatment on malarial parasites. So here you're not taking human sample, you are uh, trying to check the malarial parasite itself. When if one malarial parasite like gave one specific drug and another parasite were given with another specific drug, and what type of changes are happening inside the parasite. That's what can be studied by using 2D, even the plasma protein analysis of the virus, SARS virus, and sir talked about a little bit of challenges associated with it. So there were two combinations of proteins. So this is proteomic analysis of this specific parasite, Plasmodium falciparum. So this is the two combination of drug, artemether and lumefantrine. Both are derivatives of artemisinin. Okay. So generally, these are the drugs which are generally given worldwide if a person is suffering from malaria. So they try to check whether only one single drug is administered, what type of changes happens inside the parasite, depending upon the life cycle. So obviously, you need to take care of that, like at what phase you are taking the cultures, because it the depending upon the life cycle. The effect of that specific protein or drug will change at what stage it is. So you collected that blood sample wherein parasites will be present. You just fractionated that parasite and tried to take out. So synchronized parasites isolated from the RBCs. So from RBCs you will initially isolate parasites, and that parasites were from that parasites proteome analysis were done, except from that itself. It was solubilized by trace buffer, etc., and it was subjected to fractionation using urea, and two-dimensional electrophoresis was run. And this is just a mechanism of that. Like when this combination of drugs are given, it releases glycolytic enzymes basically. So reaction with late ring stage, that's what the stage wherein this specific drug acts. So people know the mechanism of it, and now what changes are exactly happening inside the parasite. That is what was studied in this specific uh, application or paper which sir tried to cover in this topic. So they found that when this individual drug was given, these like membrane associated calcium binding protein, heat shock proteins, etc., all those were upregulated, whereas these type of proteins, phosphoglycerate kinase, aldolase, etc., were downregulated with this drug. Whereas with this drug, all of them were upregulated, indicating they are under stress, and like heat shock protein indicating they're under stress and it is getting upregulated because it want to cover up those uh, like it want to be stable and be alive, but because of this drug treatment it is like getting to die, and it is trying to survive by upregulating this type of proteins. Whereas when combination like altered protein action levels were found, like during the drug exposure. The combination was given. These stress response proteins were upregulated, and whereas glycolytic enzymes were downregulated when the combination was given. Whereas these individuals were given at that time, this individual has upregulated that, whereas this individual has downregulated. Whereas obviously we are giving a drug treatment, so stress response will be upregulated no matter given in combination or in individual. So what did they found basically? They found that induced alteration is present depending upon the type of drug you are administering, and so they basically also established a relationship between pharmacological relevant concentration and time of exposure. So they had a pre-metadata as well, like they found that at what time they are getting exposed to that specific drug, etc. And they compared all of those free data as well and tried to check how it can affect the parasite and how quickly a person can get cured. So that was one of the application. By using two-dimensional electrophoresis, you can study that too. And in terms of SARS virus, this is how SARS virus looks like. Just an introduction of the same. You might be knowing about SARS virus because of COVID. It has spike proteins, glycoproteins, etc., as phosphoproteins as well. So similarly, healthy control as well as persons suffering from SARS. The samples were taken, all the samples were run, and two-dimensional electrophoresis were done. And they found that few of these proteins were present in the individual people suffering from SARS virus, whereas in healthy control, there was one of the protein which was there, but in like much more highly concentrated, but extensively downregulated in the person suffering from COVID. So we can, if you want to exactly find which protein is it, you can just cut this specific protein and subject to mass spec, and that's how you can study what type of protein it is. So in mass spec, I just explained when there was a doubt related to one of the topics. So basically, your sample is subjected to certain ion source where fragmentation takes place. So MS1 and MS2. So MS2, so this mass spec, mass analyzer 2. So generally they are termed as MS1 and MS2. So MS2 will give you much more accurate information of that specific protein itself. So for example, there is one specific protein 200 kilodalton, for example. So in mass spec 1, it will get fragmented initially at the first stage. So don't get confused by fragmentation of this and the initial sample preparation. The sample preparation will obviously give you, while trypsin digestion, it will give certain peptides. But those peptides, you know that you know your sequence of your protein of interest. You know what type of peptides might have been generated by trypsin digestion. So accordingly, you can subject that specific length of peptide. For example, the peptide was around uh, not 200 kilodalton, but 30 kilodalton of that specific peptide. So you can accordingly give that, like set up that specific protocol, which will only detect 30 kilodalton peptide, and that will get fragmented uniquely in mass spec one. And further, you can those specific peptides can be detected accordingly in mass spec 2 as well, where it will be fragmented further into smaller fractions, and the detector will detect those specific fragments accordingly, and it will elute according to the mass to charge ratio, and uh, it will give you those specific molecular weights and how much intensity of that specific protein is also present in your sample. Even that can be detected. So reducing condition, non-reducing condition, for example, they just gave one of the example. So in patient sample 2, so this specific protein had disulfide bonds with it. So in reducing condition, it got together, whereas it got separated here. So along with these few applications of malaria, SARS, uh, effect of drug, etc., condition A, condition B. So these were a few applications we sir talked about, but there are few challenges as well. So challenges during your sample preparation, during your analysis, If something goes wrong, your protein gel will look completely different. So this is how the gel looks if everything goes fine. Like two different examples how the gel will look if everything is fine enough. But because of certain artifact during sample preparation, during the electrophoresis, during isoelectric focusing, etc., or while doing analysis, while defining spot boundaries, while zooming in, zooming out, like cropping, etc. If you're doing something, or it depends upon user bias as well, like how you are selecting that specific spot, all of those will change because of user biasness. And there will be obviously biological and technical variation, like some manual errors while preparing the sample, or the way you have grown the culture, biological variation, technical indicating how you have collected the sample, how you have done the analysis, etc. All those will be there. So sometimes you might see, after doing everything, you might see something like this. That is possibly because of trace impurities. So your buffer which you are using for separation, the trace buffer itself has certain impurities due to which you might get streaking like this. Sometimes if you have made the gel using old timid, it might not have gotten polymerized properly. It will not get separated properly. You might end up getting a gel like this. If you have used crude sample directly because of albumin and or basically because of a lot of proteins and it is not being fractionated properly, uh, then you might end up getting a gel something like this. Then if you have not done precipitation properly, concentration, you might get a stick, something like this, and nothing will get separated as such. Uh, in terms of equilibration after isoelectric focusing, if you have only done with DTT and not with alkylating agent, some streaks like this will be observed onto the gel. While doing the analysis, while checking the image, if you are over focusing the image, because of over focusing, something like this, a gel might look like. Then the source of variation, like between individuals, like who is collecting the sample, between biological states, the way you have grown the culture, how you have collected it, etc. Effects. So this specific effects can't be controlled, whereas some can be controlled, like your sample preparation step. Like all these are in our control. You need to make sure that each and every time when you're doing collecting the sample, doing the processing of sample, you follow the exact protocol. So sources of variation, variation in sample preparation, the pH strip which you are using, the pH range, if that is different, accordingly the separation will change. If there is presence of salt in your while preparing the sample and if you have not desalted it properly, it will affect your isoelectric focusing as well as SGS page. And while separating the voltage which is applied, if it is higher, accordingly the separation uh, will change. If you are running it at a very low voltage, the separation will change because proteins will get affected depending upon the charge, right? So you need to make sure that you are using same voltage if you are trying to compare two techniques. Equilibration times, for example, if you are using 10 minutes in one technique, 20 minutes in one technique, one gel on another gel, so it will give you different results. So you need to make sure that all of these things are maintained while doing the analysis. So that's what was talked about in four lectures. And in terms of lab session lecture, I'm not exactly covering that, but Sir talked about. So he talked about how exactly, like in lab session, how exactly those gels are prepared and equilibration step is done and how analysis can be done using the software. If you have, I hope you might have gone through that lecture. I'm not sure, but that's what was talked about in lab session lecture. How that specific software can be used. So this specific, so you all can go through this. It's just 10-minute video wherein, so we can just go through it quick. So this is how the electrodes look like in terms of isolatory focusing. This is how the cups look like, wherein each and every fraction of the IPG strip of different strip length. So this is how the collector strip looks like. So after separation, this is how the gel looks. So this is the tray wherein you can do the equilibration step or rehydration step as well. So make sure you use forceps. You should not touch it with your hand, and that's how you should handle this specific gels. So you can just go through this video once in order to understand the practical aspects of the same. I think I got logged out, but okay. So each cup around 50 microliters can be loaded into that while doing rehydration or equilibration. So you need to place the electrodes while giving the voltage accordingly. So this will stop evaporation as soon as you seal it for 15 minutes incubation. And depending upon your technique, various parameters can be set up in the protocol. And so after the run, it will this light will blink, indicating that the process is done. So that's fractionation step. And once that is done, you can remove the cup and you can collect individual fractions in different append of. So basically, those peptides are collected after isoelectric focusing itself by doing this step. So this will help to get greater coverage of the protein and proteome before mass spectrometry method. It helps in reducing the complexity. And this is a software which is can be used for analysis, IQTL. So it can be done in two ways, like automatic as well as stepwise. Uh, so when you do click on automatic, it will try and detect everything, but it might miss something. So in stepwise, you can actually pick each and every spot separately and detect how much is the intensity of it, what might be the molecular weight of it, etc. All those analysis can be done using this software. So those specific bonds can be selected and can tell you about the intensity as well. So initially you can do background separation as well. Like it will only check the bands and not just the staining, which is generally visible. Like if you have not done destaining properly, there will be few stains generally visible. And this technique will just consider the band and not the background. So you can do the background separation. So this is how it is detecting each and every band here. And it is talking about that band's intensity, etc. So that is done So if you're selecting manual, you need to select each and every band separately and automatically might skip something, but it will detect most of the bands. And accordingly, data sheets of the same can be created. How much spots are there? How much is the intensity of each spot? In lane one, how many are there in lane two, etc. What might be the molecular weight of the protein? Everything can be done using this specific software. Yeah, so that's what Sir talked about in his 5 lectures. So maybe we'll take a break of 5-10 minutes and we'll start with question and answer session. So that's what the overview which I wanted to give in terms of week 3 lecture. If you have any doubts, you all can ask me, or else we can meet in 10 minutes after the break and we can start question and answer session. Okay? Yes. Okay, we'll start with the question and answer session. Okay, so this is the first question. So what Richa used two-dimensional electrophoresis to separate protein mixture and made up of four different proteins. So there was a protein mixture which had four different proteins A to D with this specific molecular weight. Following isoelectric focusing, all proteins focused on the same area as seen in the figure. So this is isoelectric focusing. So this indicated that they all had same isoelectric point. That's the reason they stopped at the same area. But further she performed SGS page of focused proteins and got 2D gel. So this is how the 2D gel look like. So you want to determine, the question is, which of the following panel depict the profile she would have received following SGS page. So as you might be knowing, SGS page is the one which separates on the basis of molecular weight. Higher molecular weight will be above and lower molecular weight will be uh, below. So the smallest is 50, which is C. So there's only one option wherein the smallest 50 kilodalton is option B. So the option should be gel B. So we can just verify it once again whether that's accurate. So 50, then D 75, then 110A, and then B 135. So it would be B A D C and B A D C. That's how it looks like. So B should be the correct answer. What could be the reason why all four proteins are focused on same? Because I just mentioned that because of the same isoelectric point. Okay, this is the next question. So Mansi performed studies on human plasma sample in an effort to find protein biomarker. She started her experiment by eliminating the sample's abundant proteins like serum albumin. Yeah, that's a good thing she has done. And she quantified and depleted her sample before preparing it for mass spec. Why did she want to deplete the sample before continuing? Because if these abundant proteins are left unchecked, the tryptic peptides will take precedence over the less common proteins, which may have more useful diagnostic properties. Because eliminating abundant proteins could significantly reduce the complexity. A second is true. There's only one option with two, one and two. Even one is two. The depletion stage may not even be necessary. No, it's not the case. Since mass spec cannot distinguish highly abundant proteins, no, that's not because one and two should be the answer. Yeah. So which of the following troubleshooting procedures? So this is how the look vertical streaking looks like. So we studied about that. So to guarantee proper protein load before IE, perform a protein assay, increase the voltage progressively while extending the time for initial low voltage phases. Utilizing in-gel rehydration, dilute the sample and load it. So what are steps need to be done in order to make sure this is not done? So I think second, third should be done for sure. Progressively increasing voltage would also help, and proper protein loading, perform a protein assay like that will help in having a proper concentration because here it was highly concentrated, whereas here it is not. So I think all three is the answer. So these are the two gels which we studied, both depleted and non-depleted sample. Depleted, non-depleted. For crude and depleted samples, the electrophoresis result displayed like this. What meaning could be drawn from these gels? So the significant abundance of proteins in the crude sample may be because of thick bands. Because the crude sample gel was improperly destained, It developed dense? No, that's not the case. Due to pure and undepleted nature of the sample, gel 1 displays greater resolution. As the sample was less complicated in the depleted sample, there were more protein spots. The fourth is right for sure. The 4 with 2 or 4 with 3. I think it's with, oh, there are more options. 4 with 1 as well. And 4 with 1 is the right answer. The significant abundance of protein in crude sample may be the cause of thick bands. Because of that thing, it was thicker band here. And fourth one, because that's obvious. If it is less complicated, it will give you more spots. What is off-gel electrophoresis related claims it's true. That's what the lab session thing. It separates proteins based on their hydrophobic interaction. It fractionates protein using a non-gel liquid phase. It is a form of two-dimensional electrophoresis, or it uses a gel matrix for protein separation. We just discussed in that video, it fractionates protein using a non-gel liquid phase. We talked about fractionation during that time. Yeah, so comparison between four proteins A, B, C, D in the control C and the sample treat. So that is control and treated of those same proteins. The panel displayed 3D images of each protein between control and treated. Respective proteins have a green border around them. So this is the respective protein, not the red one. So look, focus on the green ones. Which of the following statement is correct? In control sample, protein B and C are upregulated. B and C. No, it is not upregulated here. So protein D expression is decreased in the treated sample. Yeah, compared to the treated sample, all the proteins are downregulated in the control sample. No, not all the proteins. This is upregulated. This is upregulated. In comparison, the treatment sample, all proteins are upregulated in control sample. No, when that is not the case. So protein D expression is decreased in the treated sample. You can see it here. Which of the following occurs during first dimensional electrophoresis? So it involves SDS page separation process first dimension. So SDS is the second dimension. It incorporates molecular weight based protein separation. It employs isoelectric focusing technique which separate protein according to PI. Yeah. It involves isoelectric focusing process in which proteins are separated according to their mass and charge. It's third one wherein they are separated on the basis of their PI. That's what the first dimension of 2D is. So phosphoproteins it wanted to detect while conducting the research on breast cancer proteomics on the gel. She wants to visualize how these phosphoproteins look. So for phosphoproteins, we just studied procodiamond stain is used. Okay. So Arushi ran six gels. These are the six gels. The 3D view region made up of six 2D gels shown below. Based on the figures provided, please help her answer the following questions. What conclusion can be made by the provided figures? So gel 1, 3 and 5, these gels, gel 1, 3 and 5 may all be a part of the same batch of samples. No, this is upregulated. Like I don't think they might be of same batch of samples due to their comparable characteristics. Although we can see the comparable characteristics, there is something here which is not similar to that of 3 and 5. So in gels 2, 4, and 3, 2, 4, and 3, all spots have same intensity. No, that's not the case. Here it is highly more intense compared to this, so that's not the case. Gel 1 and 6 are technically replicates of each other. If you see the middle protein is much upregulated here, so in gel 2, 4, and 6 might belong to the same group. 2, 4, and 6, yeah, it might belong to same group or something looks similar in all of these. So 4 is there, but there is 3 and 1 as well. So gel 1 and 6 are technical replicates of each other, or just a minute, people. Okay, sorry. I think 1 can be, that's 1 and 4. So 1, 3, and 5 may be a part of, so it says may all be a part of same batch. So obviously, uh, 2 and 3 are not exactly correct if you compare the gels. 1 is possible and 4 is obviously there because it seems similar here to 4 and 6. So 1 and 4 is the answer. Yeah, I think these were the few questions which we'll discuss. I think I can also discuss few more very quickly from this course. Yeah. So in equilibration step, we do two steps. First is obviously reduction and then alkylation because then the sulfate will be available. Which of the following? Yeah, so that I can just remove that. Okay, which of the following leads to clogging of immobilized pH? So if in the pH strip, so if all of these are present, can lead to clogs. It will not run properly. We already did this phosphorylated protein procure diamond. So depending upon the strip size, the concentration of protein you can load will change. So if the strip is around 18 centimeter, you can load around 300 to 1000 micrograms. So you're dealing with acidic pI values here. of your protein. So your should be in the pH strip range should be acidic, and there's only one option here which is acidic range 3 to 6. Which of the following visualize 2D gel after the gel staining and found vertical streaks. So which of the following factor must be the possibility? Low acrylamide, percentage of low abundant protein, presence of salts. Yeah, it's because of presence of salts. What will be the position of these proteins on the gel with respect to each other if they are two proteins with same isoelectric point but differ in molecular weight? We did the first question in the earlier questionnaire. So they differ in molecular weight, so they will be adjacent to each other or vertical to each other, basically, above one above the other. Okay, so you have ran a protein in 2DE and want to compare the differentially expressed proteins. Which of the following software can be used? So this IMPF can be used. Which of the following is the correct sequence of while doing serum proteomic analysis? So you will sonicate it, you will deplete it, you will do the protein precipitation and then quantify it. Which of the following is not correct for 2DE rehydration process? Not correct. Salts are removed in the process. Mineral oil is added. Activates. So these two are done. Salts are not removed in this process. So that's the answer. So that pH gradient is made up of ampholites basically. So two-dimensional process is done. Dash degree relative to the first dimension. The first dimension is run like this. Second dimension is run like this. So it creates a 90-degree angle. So which analysis can be done when you're using spot by spot analysis? So that's spot analysis. The question itself is the answer. Which of the following is not compatible? So we just studied that. It was silver stain which is not compatible. The reaction is stopped by adding acetic acid. Is correct about isoelectric focusing. Proteins are resolved on the basis of pH. Yes. Involves the use of immobilized pH. Yes. Presence of salt interfere. Yes. All of the above. So image quant software is true. It can remove background. Yeah, it can be used for quantitative. It employs standards for molecular studies. Yeah, all of the above. Then there are two steps, automatic and stepwise. We just talked about that. But which method is preferred? So stepwise is preferred, but you can take each and every band because of stepwise, as automatic skips some bands. Cannot be performed by this software. Dye analysis is another technique. Only 2D electrophoresis, colony counting, array analysis can be done. So dyes cannot be done by this software. If you run an SGS page, you are using this software which you not give desired results. What is the option in software? So you are wrongly selected one of the following parameter. So it didn't give you the desired results you wanted. So it is possibly because of your selected fixed bandwidth. Automatic will help you detect the give you much more better results. So it's as we are talking about differently expressed proteins. So fixed bandwidth because of which you did not give desired results. Yeah. So these were the few questions. Then so next week we can meet and we'll be talking about DIGE as one more gel-based proteomics assay. Uh, so accordingly we can talk about that in the next week. Thank you for attending iSeries initially. Then you can go through this question and answer session in the recorded lecture. And thank you. I can end the lecture here. Thanks.